Hello, I'm Laura Marshall.
And I'm Melinda Rose, and this is Light Matters for October 17th, 2012.
On this week's show, Melinda speaks with a pioneer in the field of silk optics. I share news from the Neuroscience show, and a photonic gel is found to be a metamaterial with many uses.
Frontiers in Optics, the Optical Society's annual meeting, is taking place in Rochester, New York this week. One of the hot topics at the show was the use of silk, both from silkworms and spiders, in optics applications. I spoke with Fiorenzo Omenetto of Tufts University, who pioneered the field of silk optics, about the work his lab is doing to use silk as a material for photonics and high technology applications.
I think that in fact, some of the nice applications that we are working on have joined not only optics but also electronics. So it's a combination of materials that can be implanted and then gradually resorb over time and disappear. So the things that we're working on are, for example, implantable heaters that come from a combination of, say, a wireless receiving coil that is built on a film of silk that then goes under the skin and then heats up on command and mitigates infection at its implantation site. By the same token, right now we're working also on optical relay systems. So, for example, on reflectors, silk reflectors that can be implanted and then can send back more information, more optical information when they're left under the skin, for example, and then interrogated from the outside.
So, do you have any examples of prototypes there that you could show us, perhaps?
The thing that I always show is an old diffractive optics card there was an old card that actually was distributed at trade shows at Clio, in fact, and so you can see here a bunch of micro lenses and phase masks. And this is an old digital optics polycarbonate card, but this is 100% silk. So this has been shown for the past couple of years. It has a lot of thumbprints, but basically it comes from this. So from our cocoons.
His work is so interesting. What else did you talk about?
Well, there's lots more that we don't have time here to show, but you can see our entire interview, including Dr. Omenetto's culinary tips for silk, at photonics.com/lightmatters.
Wait, seriously, so you can eat this stuff?
Yeah, if you were really, really, really hungry. So anyway, that's how I kept busy this week while you were in the Big Easy. What was the big news at the Neuroscience show this week?
Well, the conference was just great. I really wish I could have spent more time and more lectures, but what I did get to see while I was there was so interesting. Next year, I think they're going to have to hold the Optogenetics Symposium in the biggest meeting room they can find. Optogenetics is just getting more and more popular every year. This year, the room was so overcrowded that the line to get in extended from the doorway all the way back down the hall and back again. It really was standing room only by the time I got in. In that particular symposium, Mark Schnitzer of Stanford talked about his group's work on fluorescence microendoscopy in not only living but awake and behaving mice. He described two methods, epifluorescence, which uses a single photon, and laser scanning, which uses two. In his group, they've been mapping neural circuitry in the mammalian brain, including the hippocampus and neocortex, and they've actually achieved time-lapse imaging of cells deep in the brain thanks to their approaches. Schnitzer said you can image cells in behaving mice either by imaging alert head-restrained mice so that they can't behave freely, or by using mini microscopy in which the mice wear little hats with the microendoscopes attached. Restraining the head gives better resolution and contrast, he said, but the little hat method allows them to take images of the mice as they go about their lives. And the results are fascinating. For example, they can now decode where the mouse is in space in its little container just by looking at the neural images. Carl Dijsselroth, also of Stanford, was the final speaker, and he talked about his group's work in optogenetic loss of function in mammalian behavior. With their work, which requires no chemicals, they can achieve non-invasive control of deep brain targets, and they've been studying the circuit dynamics of neuropsychiatric disorders, for example, anxiety. He also described their clarity method, which can get brain-wide images with no sectioning, and he just won the NIH Transformative Research Award for this approach. And he says it could someday elucidate brain circuitry abnormalities involved in psychiatric diseases like depression, PTSD, drug abuse, autism, and schizophrenia. In the afternoon, I got to sit in on a lecture by Stephen Hyman of Harvard. His talk, geared towards neuroscience students, was about how to navigate the changing global neuroscience ecosystem. By that, he mostly meant how to deal with the possibility of funding cuts for the sciences, the current state of industry, the occurrence of fraud, and the pressure to publish only positive results. He called on students to develop large-scale, interdisciplinary, collaborative approaches to advocate for basic science and science in general, to rethink animal models, and more. It was an illuminating and inspiring talk, and I think the full house agreed. The exhibition show floor was hopping as well, and I got to check in with Olympus and Photometrics and Q Imaging and a bunch of other photonics companies. They basically said that the leads this year were high quality, and the show, in general, they said the show was really great.
Great. I'm sure we'll see some of what you learned informing the next year of Biophotonics magazine.
Definitely.
Finally today, a photonic gel developed at Rice University and MIT has layers that can be tuned to reflect specific colors when exposed to particular chemicals. The thin films are incredibly cheap to make and may inform future multiband optical elements in laser-driven systems or high-contrast displays, among other applications. The micron-thick material combines polymers into a unique self-assembled metamaterial that changes color when exposed to ions in a solution or in the environment based on the ion's ability to infiltrate the water-loving layers. The work appeared in ACS Nano.
Well, that's it for this edition of Light Matters, Photonics Media's weekly newscast. We'd like to hear from you. Tell us what you like or don't like about our program. We welcome comments and suggestions at lightmatters@photonics.com.
You'll find links to share and subscribe to Light Matters by clicking the Share with Friends button on our video player.
Thanks for watching, and we'll see you next week.